Okay. Quick note, if anybody is interested in the project that was the title in the pamphlet, ask me about it and I'll give you the Cliff Notes. So that was another research project that I presented at a different symposium. Today I'll be talking about how does the above-ground fungal endophyte Epichloë coenophiala impact colonization of arbuscular mycorrhizal fungi in tall fescue. Plants, as you know, have many, sometimes very complex interactions with microorganisms, both above and below ground with things like viruses, bacteria, and fungi. Grasses, for example, commonly form association with fungal symbionts, some of which are host-specific, and many of these behave as obligate biotrophs, meaning that they must gain photosynthetically fixed carbon from their host plant in addition to requiring their host plant in order to complete their life cycle. When this association is a mutualistic one, they can have many positive effects on plant fitness, both above and below ground, such as increased stress tolerance or plant growth. While we know more and more about the individual effects of these fungal symbionts on their plants, we know very little about how multiple symbionts within the same host plant interact with each other or how those relationships are mediated by the host plant. One plant fungal symbiosis that is of particular interest to us is that between tall fescue, a common cool season forage grass in the southeastern U.S., which you've heard about a little bit today, and Epichloë coenophiala, an above-ground asexual fungal endophyte. This is often termed a defensive mutualism, whereby in exchange for photosynthate and emotive reproduction from tall fescue, the endophyte produces alkaloid compounds that deter herbivory and increase plant fitness. Unfortunately, these ergot alkaloids produced by common toxic endophyte strains, or CTE, harm livestock animals through things like reduced reproductive success or impaired heat tolerance. In response to this, novel non-toxic endophytes have been isolated from wild populations of the same endophyte species, which do not produce the ergot alkaloids that deter mammalian herbivory, but still retain production of the lowline alkaloids, which deter insect herbivory. These have been developed for seeds, for commercial seeds for pasture use, whereby seeds or plants with the common toxic endophyte will be treated to remove the endophyte and then re-inoculated with a novel endophyte. This is very important because now that we can manipulate which types of endophyte are in which types of tall fescue, not only can we evaluate how the presence of this endophyte impacts plants and other organisms, but how genetic variation influences these relationships. Another widespread and very important fungal symbiosis that we're interested in is that of arbuscular mycorrhizal fungi, or AMF. These are reported to colonize about 80% of terrestrial plants. They're not as host-specific as these fungal endophytes. But they're also reported to consume about 20% of terrestrial photosynthate products. So they have large ecosystem implications for carbon cycling in addition to soil chemical and physical properties. The plant relationships with AMF are generally, but not always, a mutualistic one. So they're good for increasing water and nutrient uptake, such as phosphorus, especially when these resources are scarce. They're also good for enhancing soil properties, such as aggregate stability and size distribution. So this makes them important players in pasture productivity and sustainability. Some prior research in greenhouse studies have investigated the impact specifically between common toxic endophyte infected tall fescue and AMF. But they found that CTE plus tall fescue decreases inoculated AMF abundance and spore relation, sometimes even in other plants or in the surrounding soil. A possible mechanism for this that was demonstrated in perennial ryegrass, which hosts a similar Epichloë endophyte, was that endophyte infection may actually compete with AMF for plant carbon resources. However, in field studies of other cool season grasses, some of them native, and their associated Epichloë endophytes, have found that endophyte infection actually stimulates arbuscular mycorrhizal colonization and may even have additive effects on plant growth in addition to the benefits gained from the endophyte. And we still know very little about how endophyte strain plays into this. So what are the novel endophytes affecting in this relationship? With that, our objectives for this study was to evaluate how both the presence and the strain of Epichloë coenophiala in tall fescue impacts the relationship between the fescue, the endophyte, and AMF, and how that impacts root colonization and plant nutrient concentrations. Hypothesizing, for example, that CTE plus tall fescue will have lower root colonization rates than endophyte-free or novel endophyte-infected tall fescue. I'm also interested in how the resulting differences impact soil properties, so how fescue, endophyte, and AMF associations change aggregate size and stability, hypothesizing that higher AMF colonization rates will increase size and stability of water-soluble soil aggregates. The study was conducted at the University of Kentucky Spindletop Research Farm in Lexington, Kentucky. I have 2 sites from this study in my whole project, but I'll be talking about one of these today. These are the SCA plots, which is a randomized complete blocking design with 6 blocks and 4 endophyte treatments. These are endophyte-free, E-, common toxic endophyte infected, CTE+, or infected with one of 2 novel endophytes, AR542NE+, or AR584NE+. And all of these are in a PDF variety or pasture demonstration farm variety of tall fescue provided by the Noble Foundation and established here in 2008. In late May of 2013, we dug 3 individual tall fescue ramets from different plants in each of my treatment plots, and also collected and composited 3 soil cores from the upper 10 centimeters from each of my plots. All of these were stored in the freezer at -20 degrees to await analyses. For my first objective regarding my plant characteristics, I first verified endophyte infection in individual tillers from these ramets using an Epichloë protein-specific immunoblot assay. So anything that was endophyte infected when it was supposed to be endophyte-free, or endophyte-free when it was supposed to be endophyte infected, didn't make it into my statistical analyses. I threw those out, and not very many of them happened to be that way. I also assessed mycorrhizal colonization as a percentage in associated roots using a microscopy method where I stained the roots, laid them out on microscope slides, and then looked for infection structures of the arbuscular mycorrhizal fungi, things like arbuscules, vesicles, and hyphae. I also quantified nitrogen phosphorus concentrations and separated root and shoot tissue via a wet digestion method. For my second objective, my soil properties, using plot composited soil cores, I determined water-stable soil aggregate percentages using a wet sieving method. I actually separated these into microaggregates, anything greater than 53 microns, and small macroaggregates, so anything greater than 250 microns. My statistical analyses incorporated a Proc mix model in SAS for a randomized complete blocking design with endophyte treatment as a fixed effect. I also took the opportunity to run some regression analyses using AMF as an independent variable with each of my measurements. These are the results of my AMF infection percentage. On the y-axis is my AMF colonization rates. So how many total times I viewed that slide whenever I saw an infection structure, that's how I calculated those rates. And then across the x-axis are my endophyte treatments. You can see there was no significant effect of endophyte treatment here. Very similar rates, very high rates of AMF colonization across the board for my endophyte treatments. Although you'll notice that there is a slight difference, I did see the pattern of separation that I expected to see, especially between the endophyte-free and the common toxic affected. Not significant, but put a pin in that. These are my root nitrogen and phosphorus as a percentage. So green tones are nitrogen, red tones are phosphorus. On the y-axis is just the percent root nitrogen or percent root phosphorus. Again, there was no significant endophyte effect. There was a little bit of separation, but not pretty negligible. These are my shoot nitrogen and phosphorus. So again, green tones, nitrogen, red tones, phosphorus as a percentage in the separated shoot material. There was no endophyte effect in this. There was a very weak trend here in the endophyte treatments for shoot nitrogen, and this was interesting because it was driven by this large effect, or by this difference between the endophyte-free samples and the common toxic endophyte-infected samples, the CTE+. And while this wasn't terribly significant, it is at least consistent with other data that I collected for another project for this, where there was that difference in shoot nitrogen between the endophyte-free and the CTE+ plots. So not significant, but it does show up across multiple years. Those was in 2011. And there was no endophyte effect on shoot phosphorus. The only measurement that I found significantly correlated with AMF colonization rates was tall fescue shoot phosphorus. And while there was a very low, the actual correlation coefficient was rather low, you can see that as shoots were more deficient in phosphorus, they were relying a little bit more heavily on those AMF root colonization rates. And this is what you would expect to see and is supported by other studies. Possibly because my differences in AMF colonization rates were so subtle, I didn't see any significant differences in my water-stable soil aggregate percentages, either in microaggregates or in the macroaggregates. So there are no endophyte effects on any of those either, even though we're very, very good distribution of macroaggregates here. So far, we have little support for my hypotheses that there would be endophyte-mediated differences in AMF colonization or in soil properties. So no endophyte effects across the board there. but I still have to do this analysis at my second site, which is still at the Spindletop Research Farm. It has matching endophyte treatments, but this time incorporating multiple tall fescue cultivars. And it's interesting to note that some researchers from the University of Colorado went ahead and took some rhizosphere soil from my second site and sequenced the DNA to look at microbial diversity. And they found that in the rhizosphere, around common toxic endophyte infected and the novel endophyte treatments, there was increased abundance of AMF DNA. So I'll be interested to see how that pairs with what I'm looking at, even though they're measuring different things, DNA in the rhizosphere versus me actually quantifying, infection in the roots. So I'm eagerly awaiting what I get for that. However, right now, I think I can safely say that different Epichloë endophyte strains and presence may not alter AMF colonization rates or plant nutrients in tall fescue, which is good news for selection of novel endophytes or pasture sustainability. Many thanks to my major advisor, Dr. Rebecca McCulley, and to my PhD committee and my lab group who helped me sample. And I'd love to take your questions now. Yes, sir.
Could it be the lack of differences among your treatments due to the fact that there was very little AMF inoculum?
Okay.
These soils, or did you inoculate?
I did not inoculate. So these were field soils. And actually, because it was a field soil, there could have been maybe 10 different species out there available to be colonizing these. And the rates that I found, up to 40% or so, they're actually quite high. In the greenhouse studies that I referenced earlier, they were lucky to make it up to 10%. Yes?
And along those same lines, I did some work with Spindletop, and that soil is fairly high in phosphorus.
Yes, it is.
Phosphorus levels high in the soil usually are not at the same time we have high levels of AMF.
That's correct, yes.
Do you think that might also be contributing to this?
Those are my suspicions exactly. So what he's talking about is that generally you see a high negative correlation of soil phosphorus with AMF colonization rates. So when you have very high levels of soil phosphorus, you have very or somewhat low levels of AMF colonization because they're not needing to rely as much on the AMF to excavate any of that phosphorus. And so part of the reason that I could have found very low differences in my endophyte treatments of their AMF colonization rates was because my site is so high in phosphorus naturally because of our phosphatic limestone material. So yes, that's true. And you saw a little bit of that effect with my shoot phosphorus. And I haven't sent off my soil samples to have soil phosphorus done yet, but that is definitely in the works. So that should help me, kind of get to the bottom of that a little bit more.
You need a low soil phosphorus site in Kentucky. There's one in Hopkins County on Hancock Farm.
I'll keep that in mind. Thank you.